To kick things off, just think about the number 60. It takes just 60 seconds, literally 1 single minute, for a tiny cellular machine called a ribosome to synthesize an average-sized protein made up of 300 amino acids. I mean, think about that. Every minute of every day, your cells, and the cells of pretty much every living thing around you, are snapping together these incredibly complex molecular chains. And they're doing it with a speed and precision that modern human engineering can honestly only dream of.
So, let's start with Section 1, the universal protein machinery.
Because across all kingdoms of life, whether you're a microscopic bacteria or, a human being, this machinery universally relies on 3 main types of RNA. First step, we've got mRNA, or messenger RNA. Think of this as the actual blueprint, directly copied right from the DNA. Next is tRNA, the transfer RNA. This one's absolutely crucial. It acts as the adapter. It essentially reads the genetic code on that blueprint and hauls the exact right amino acid over to the assembly line. And finally, rRNA, or ribosomal RNA. This combines with proteins to build the ribosome itself, which is the actual factory floor where the magic happens.
Now, this factory floor operates in 3 core steps. Step 1 is initiation, where the small ribosomal subunit binds to the mRNA. And what's kind of fascinating here is that this first step is actually the rate-limiting step for everyone. Basically, getting the factory started is the hardest part. Once it's initiated, we hit step 2, elongation. This is where the tRNA moves along the mRNA, rapidly forming peptide bonds between those amino acids. And finally, step 3 is termination. A specific stop codon enters the ribosome, acting like a giant red light. Since there aren't any tRNAs for a stop codon, it just triggers the release of the newly built free-floating protein.
Moving on to Section 2, the core contrast, prokaryotes versus eukaryotes.
This is where things get really interesting, right at this fundamental divide. On one side, we have prokaryotes, like bacteria. They don't have a nuclear membrane, and they don't have internal organelles. They are the ultimate open-plan factory. Literally everything happens in one giant room. On the other side, we have eukaryotes, like animal and plant cells. These feature a rigid nuclear membrane and complex membrane-bound organelles. They're totally compartmentalized, with highly specialized departments separated by literal walls.
Which brings us to Section 3, bacteria, fast and coupled. Okay, so the secret to bacterial speed is the concept of coupled transcription and translation. Since bacteria don't have a nuclear wall separating their DNA from the rest of the cell, well, there's no waiting around. The exact moment the DNA blueprint starts being transcribed into mRNA, multiple ribosomes just immediately jump onto that freshly made RNA strand and start building proteins. The blueprint is literally being read while it's still being printed. It's a simultaneously coupled process, and that's what allows for their incredibly explosive response times.
And the crazy thing is, bacterial efficiency doesn't even stop there. Bacteria also use what's called polycistronic mRNA. Now, unlike our cells, where one blueprint usually means one specific protein, bacteria can print a single continuous strand of messenger RNA that contains the code for multiple totally different proteins all at once. So say a bacteria suddenly needs to synthesize an amino acid like tryptophan just to survive. It'll just print one continuous polycistronic blueprint, and ribosomes will simultaneously manufacture every single enzyme needed for the job in one fell swoop. Boom. Done.
All right, Section 4, eukaryotes, separated by design.
Now, in stark contrast to that chaotic speed of bacteria, eukaryotic cells are all about strict compartmentalized control. For us, it's a highly regulated 2-step process. Step 1 happens exclusively inside the nucleus, where the mRNA is transcribed from the DNA, but it can't be used yet. Step 2 requires this newly minted RNA to be processed. It has to get a protective 5' cap and a poly-A tail added to it. And only after passing these quality checks is it finally allowed to be exported out of the nucleus into the cytoplasm, where the ribosomes are actually waiting to start translation. And while the overarching process is highly conserved between both domains of life, the specific parts on the assembly line do differ slightly. For instance, during the elongation phase, bacteria use a helper molecule called elongation factor Tu, whereas eukaryotes use an equivalent called elongation factor 1A. And when it comes to finally stopping the assembly line, bacteria rely on 2 different release factors, RF1 and RF2, to read their stop codons. Eukaryotes, though, are actually a bit more streamlined at the finish line. They use just one universal release factor that recognizes all the stop codons. Let's get into Section 5, eukaryotic targeting and secretion. So here's a huge question. If your eukaryotic factory has all these different specialized rooms and organelles, how does a newly made protein actually know where to go? Interestingly enough, regardless of its final destination, whether it's meant to stay inside the cell or be secreted outside, every eukaryotic protein destined for secretion starts its life exactly the same way. It begins synthesis on a free-floating ribosome right in the cytosol. But then, it kicks off this beautifully coordinated mechanical sequence. As that free ribosome starts building the protein, the very first part of the peptide chain emerges, and it acts as a literal shipping label. A special molecule called a signal recognition particle, or SRP, spots this label and binds right to it. And the moment it binds, the SRP acts like a handbrake. Protein synthesis is completely, momentarily halted. The entire factory line just stops.
Now, why does it stop? So the package can actually be delivered to the right department. The SRP drags the whole paused ribosome over to the endoplasmic reticulum, or the ER, and docks it onto a receptor complex on the ER membrane. Once it's securely docked, the SRP releases the handbrake and takes off. Synthesis resumes, but now that growing protein is being pushed directly through a pore into the inside of the ER. A special enzyme snips off that initial shipping label, and the newly completed protein safely folds inside the organelle, totally ready to be secreted. It is just an astonishingly elegant solution to the whole problem of compartmentalization.
And this perfectly highlights the difference in how these two factories are managed. Bacterial regulation is brilliantly simple and reactive. They hate wasting energy, so they primarily control things right at the transcription level. A great example of this is the SOS response. If bacterial DNA gets damaged, sensor proteins immediately trigger the simultaneous production of over 40 different repair proteins. Eukaryotes, on the other hand, have incredibly complex regulation at every single checkpoint. They regulate at the initial transcription, during the transport of the mRNA through the nuclear wall, by controlling how stable the mRNA is, and finally, down at the translation level itself. So this really leaves us with one final fascinating question. We've just seen two completely different ways to solve the exact same biological problem. Which evolutionary trade-off actually wins out? Is it the rapid, chaotic, open-plan efficiency of bacteria, allowing them to adapt to environmental stress in mere seconds? Or is it the highly controlled, strictly compartmentalized precision of eukaryotes, which is what allows for the incredible complexity of multicellular life like us? They are both absolute masterpieces of natural engineering. Think about that evolutionary arms race the next time you marvel at the microscopic world. Thanks so much for joining me on this explainer, and I'll catch you next time.